Okay, good morning everyone and welcome to the 2016 Royal Tyrrell Museum Speaker Series. Today, February 12th, is Darwin Day, a special celebration commemorating the birth of the famous naturalist Charles Darwin. Oh, he's up there. There you go. Thank you. The author of the book on the origins of species, who was born 207 years ago. So happy birthday, Charlie. To celebrate Darwin Day, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Mary Silcox. Mary is an associate professor in the Department of Anthropology at the University of Toronto in Scarborough. Mary is originally from Mississauga, Ontario, just outside of Toronto. She obtained her bachelor's degree in anthropology and zoology at the University of Toronto and subsequently moved to Baltimore, Maryland to pursue a PhD in functional anatomy and evolution at the Johns Hopkins University School of Medicine. For her dissertation, Mary studied the relationships of Plesiadapiformes, a group of small primitive mammals closely related to primates that is today extinct. Following her PhD, Mary held a 2-year postdoctoral fellowship position at the Pennsylvania State University, became an associate professor at the University of Winnipeg for 8 years, and finally moved to the University of Toronto Scarborough in 2010. Mary's research interests focus primarily on the origin and evolution of primates and of their close relatives. Her work consists in studying the relationships of primates in the greater context of all mammals, and in looking at the evolution of particular characteristics such as large brain size and grasping, which have been thought to be distinctive features of primates. Today, Mary will talk about the early evolution of brain in primates based on the fossil record. So without further delay, I present you Dr. Mary Silcox.
So Francois stole my thunder a little bit, but I did just want to remind everyone that today is Darwin Day, and we really all should be on holiday today if some of us who believe this should be an international holiday had their way. There is, of course, an enormous diversity of modern primates. We have primitive things like this ring-tailed lemur and galago, monkeys, and, of course, apes. And it's somewhat challenging to come up with characteristics that are distinctive of all of this group because of this great diversity. But one of the features that has often been cited is relatively large brain size. So as an order, on average, primates have relatively large brains, with, of course, humans being the most extreme in this. We have the largest brain relative to our body size of any mammal that has ever been studied. And so this is one of the traits that has been keyed upon as something that requires explanation in the study of primate evolution. Why is it that primates have these very large brains? Now, this has been studied in the context of the diversity of living primates. There's actually an extensive literature that's looked at it from that perspective. To the extent that when I was on sabbatical, the last time I was on sabbatical, I was basically retraining myself in the study of brain evolution. And every time I went into my office, I was in danger of being crushed under the piles of paper on the evolution of the brain, because there's a lot of interest in this topic. But what has been studied much less is the evolutionary history of this from the perspective of the fossil record. And there's a reason why that's important. So this is a graph modified from the work of a fellow named Harry Jarrison, who was one of the first people to study the evolution of the brain. And what this shows is that there's a strong relationship between brain size and body weight. So these things have always been strongly related. But the nature of this relationship has changed through time. So if we look at modern animals, for a given body weight, you would actually expect them to have a larger brain than one from, say, the Oligocene or one from the Paleocene. So there is a temporal effect in brain size. And that means if we have information on brain size in some fossil, it's actually quite problematic to try and compare that to the modern record of mammals, because you wouldn't expect an ancient fossil animal to have a brain of the same size. Now, we're not really sure why this relationship between time and brain size exists. It might have to do with predator-prey dynamics. Basically, every time a prey animal gets smarter, the predators need to get smarter, and then the prey need to get smarter, and the predators need to get smarter. And basically, everything is either predator or prey, right? But it's actually kind of an understudied question why we have this temporal effect. It's really more just an observation. The other reason why we need to go to the fossil record is just because primates have been evolving away from their common ancestor for at least 65 million years, possibly longer if we believe people who study DNA. And that's a heck of a long time for the early events associated with brain size evolution or brain evolution to be overwritten by subsequent changes. So the fossil record becomes very important if we want to ask the question, why did primates originally start to get a big brain? Now, of course, the problem is that brains don't fossilize. And until about 20 years ago, you only had really two options if you wanted to study the form of the brain. In both cases, it involved studying the internal aspect of the skull. So there's quite a close relationship between the inside of the skull and the brain. And in fact, this is even stronger in small mammals than it is in larger mammals. There's actually a tighter fit, which means that if you have some way of representing the inside of the skull, you actually have a fairly good representation of what the brain looked like. This is an example of what's called a natural endocast. So this is a fossil hominin, very famous fossil hominin called the Tong child. And it was preserved with a partial natural endocast. So this thing that looks like a brain is where the skull filled up with matrix and then the skull itself cracked away. And we ended up with this sort of blobby-looking thing that's sort of vaguely brain-like. And alternatively, if you didn't get preservation of a natural endocast, if you had a fossil skull that was hollow, you could actually pour latex inside, kind of squish it around, then pull the latex out of the foramen magnum, the big hole in the base of the skull, and produce a fairly good representation of what the inside of that skull looked like. The problem is, the animals I study have skulls that are maybe at most 5 or 6 centimeters long. They're very fragile. They're often at least partly filled with rock, and no curator in the world is going to let you pour latex into them and squish it around and try and pull it out, because you'll destroy the skull. So in the last, let's say, 20 years, a new approach has become sort of the standard for trying to study this, which is to use high-resolution CT or CAT scanning data. And I always like to show this picture because it looks like real science. Ooh, it looks like something out of, Dr. Frankenstein's lab. This is actually a micro-CT scanner at Penn State University, which I used when I was a postdoc. Basically, you have the specimen actually sat on this pedestal. This is a very high-energy X-ray beam and then a receiver. And this lets you get much higher resolution CAT scan data than you would get from, say, a medical CAT scanner, which is important when you're studying really small animals. We actually don't use this machine anymore. The machine we use now is much less impressive looking, but it's at Duke. But this one actually makes me look like I'm a real scientist, so it makes me feel warm and fuzzy. So this is just one example. This is one of the critters that we study. This is an animal called Ignatius. A specimen's about 54 million years old. And just take a look at the scale here. This whole thing is about 5 centimeters long. This is one cross-section through that specimen. And you can see this is where the brain would have been. It is partly filled with matrix here. But you can tell the difference pretty clearly between matrix and bone. So what we did is for all 821 slices in the data set where the brain would have been, we went through and filled this with pure white. Well, by we, I mean, I actually did the first version of this one, but it's pretty ugly. I had a student who then went in and did it named Claire Dahlman, and took like a year and a half at that point. And we stacked it all together and made something that looks a lot like a brain. And this actually even has preservation of the traces of things like meningeal vessels, so these would have been blood vessels on the surface of the brain. So this gives us an opportunity to study not only the size, but actually the form of the brain and things that go fairly far back in time. Okay, so these are the three main questions that I'm trying to answer. The first one is kind of the holy grail in terms of the study of brain size evolution in primates, which is just the big why question. Why is it that on average primates have large brains? And I'm not going to give you a complete answer to that question today. I don't think we know a complete answer to that question, but I'll give you a partial answer to that question today. I would argue that the second question is absolutely critical if we want to try and answer the first question. So if we want to answer the why question, we also need to understand the when question, because we need to understand the evolutionary context in which these changes are happening if we really want to understand them. And then finally, all of this depends on knowing what's primitive for the primate brain, because until we know what's primitive, we're not in a position to understand what really is actually special and different about the primate brain. So this is a huge part of what we're trying to do, is to understand what actually the relevant comparison is for comparing, for example, early primates. Now, all of this depends on having a hypothesis of relationships. And I'm just going to walk you through this tree. I know it's not the most exciting diagram, but there's basically 3 points in this tree that I'm going to talk about today. And I want to talk to you about how they kind of are related to one another. So this is an analysis that we published in 2010, but it's actually largely concurrent with hypotheses from DNA. So people believe we might be right, which is always a nice change. So first off, we have the group that includes all living primates. So all of these guys, even him, would be a euprimate. But it also includes fossil members of these living groups. So these are things that we can very clearly tie to living primate groups. So for example, living prosimians like lemurs have a tooth comb. This is a fossil form that has the same kind of morphology, clearly related to those guys. Modern apes tend to have very long arms. This is a fossil ape that already has long arms for four-limb dominated locomotion. So nobody argues about whether these things are primates. So we put them in this group called euprimates, which really translates into things that nobody argues about. Now, my colleagues and I include a group of fossil animals also in primates, which are not clearly related to any modern group. And this is a cluster of things that are more closely related to living primates than to anything else, but are not direct ancestors to anything that's alive today. They're what are called stem primates. And there's actually a lot of them. There are about 140 named species. There are actually a number of unnamed species. Dr. Scott and I were just looking at some beautiful unnamed species that he has. Very diverse group. There are 11 distinct families. They range from things that are almost as primitive as some primitive mammals. So purgatorids are quite primitive in terms of their morphology, to things that are really quite specialized. So for example, this is a Saxon Aled. It has this weird kind of mitten-shaped tooth. This group actually evolved the same type of morphology in a different locus. Wonderful diversity of different forms, particularly in dental anatomy. And they appear about the time the dinosaurs are going extinct, so right around 65 million years approximately. There's a bit of debate about exactly how old they go. And they last for quite a long time, actually, something like 20 million years, and are found across Laurasia, so North America, Europe, and Asia. There's records of these guys. Now, a lot of what we know about Plesidapiforms comes from teeth, but there are starting to be a better record of other parts of the skeleton. And this largely comes out of the work of, first off, a fellow named Peter Hood, but subsequently a fellow named John Block. And this is material that came out of limestone blocks. So they went out into the field and they brought back these big blocks of limestone and then used acid to prepare the rock away. And what you end up in, at least some cases, with is actually fairly complete skeletal specimens. And the really nice thing about this is you actually get preservation of even relatively small parts. So this, for example, is a species we named in 2007, Dryomomesolei. This is an animal that would have weighed about 30 to 40 grams. So an animal that would have quite comfortably perched in your hand. And these are wrist elements, each of which is actually smaller than a grain of rice. So they're kind of incredible fossils to find. Now, the good news from the point of view of those of us who are interested in brain evolution is, along with these skeletons, at least in some cases, we are starting to get reasonably preserved skulls as well. And that allows us to start to actually look at the fossil record of the evolution of the brain in this group of intermediate primates. And then finally, we're also interested in things that are a little bit more distantly related. So primates are part of a group with two other orders of mammals that are still alive today. These are the Dermopterans. This is a group of gliding animals from Southeast Asia. They're sometimes called flying lemurs, even though they don't fly and they're not lemurs. So they are, in fact, non-flying non-lemurs. They're gliding non-lemur animals. And they are, what actually they mostly look like is broken umbrellas. They sort of hang from a tree like a broken umbrella all day. And then these guys, these are so-called tree shrews, again from Southeast Asia. They look a little rodenty, but they're not actually rodents. They're more closely related to primates than they are to rodents. And they are, although they're called tree shrews, they're not shrews, and most of them don't live in the trees. So anyway, they're small furry animals that scamper around, sometimes going in the trees, but spending a lot of time on the ground as well. Anyway, these would be the ideal animals to look at to study what is primitive in primates because they're the things that are most closely related, at least in terms of modern orders. The problem is that they effectively have no fossil record. I mean, there are teeth. That's unfair. They have some fossil record. They just don't have heads. So for those of us who like heads, they don't, So that means we really can't study the evolutionary history. Now, I actually have a student who's just going to start studying modern Scandensian endocasts because we actually don't know that much about them. But we're always going to run into that problem of trying to compare modern animals to fossil animals when we know there's a temporal effect in brain size. So the next group out to Euarchonta is actually this group called Galeries, which includes rodents and rabbits. And as it happens, Galeries actually has a pretty fantastic fossil record. So one of the things we've been starting to do is to look at Galeries as potentially giving us another line of evidence for what's primitive to primates because they're actually only a few nodes removed. So these are the 3 points that I'm going to basically be talking about in my discussion today in terms of what we know about the fossil record of brain evolution. Okay, so let's start with euprimates. There's actually a pretty decent record of natural endocasts. So again, those are endocasts that form without you having to do any fancy CT scanning. They're just basically, they form in the skull, which was described in the 1970s and 80s. So we have some idea from that what brains in these early euprimates look like. And these are animals that start to appear about 55 million years ago. We're just starting to do virtual endocasts for some of these guys. There are 2 that are published, but they weren't published by my research group, so I'm going to show you these pictures instead. This is actually work that was done by a student working with me at the University of Florida. She's now at Duke. And she's done endocasts for basically all of the skulls for a group called Adipoidea that are quite well preserved. So this is actually quite an amazing new source of information on what these early euprimate brains look like. So first question, of course, we have to ask is how big were they? Right? We always, when we're talking about brains, we always have to talk about size. But the problem is absolute size doesn't tell you very much. So in terms of absolute size, elephants have bigger brains than us. And I don't think that elephants are smarter than us. I mean, maybe they are and they're just not letting on. But in terms of the things we're interested in, absolute size, because it's so strongly related to body size, doesn't actually give us what we're looking for. Now, you might think, okay, well, then I'll just take a ratio of brain size to body size. That will give me something that will be relevant. The problem with that is that small animals have, for their body size, bigger brains than larger animals do pretty consistently. So if you just do a simple ratio, actually mice seem to have bigger brains than we do. And again, I don't think mice are smarter than we are, right? So in terms of trying to get at kind of what we're interested in, Harry Jarrison proposed this metric, which is the actual brain size over what you would expect for an animal of a particular body mass. So a value of 1 is going to be just a completely typical mammalian brain, just what you would expect. Okay? And that's helpful because it's also something you can kind of conceptualize and understand. So here we have some information on, here's living primates. I actually took humans out of this because we screw everything up. We're like over here. Our EQ is about 5 to 6, so we have brains about 5 to 6 times what you would expect for a typical mammal. We're very smart. But actually, most extant primates, most living primates are pretty smart. So here's 1, and you can see that most living primates have EQs of greater than 1. And of course, this isn't news. We know that primates generally have pretty big brains. Here we have fossil primates, fossil euprimates, and you can see that really they're not that impressive. A lot of them actually have EQs of less than 1. And this reflects that fact that there is this temporal effect in brain size. So again, you wouldn't necessarily expect a fossil euprimate from, say, 48 million years ago to have a brain the same size as a living primate because we know that brains in general have gotten larger in mammals. However, the question is, what were they like in their time period? Were they the smart kids on the block or were they, pinheads? Well, this is a terribly messy sample of basically what we have of other things from around the same time period. And what you can see is, and this is not really my point, it's really actually Harry Jarrison's point, but fossil euprimates have always been kind of exceptional for their brain size, for their time period. It just is, if you do the modern comparison, they don't seem that impressive, but they were the smart kids on the block when they were alive. And if we take a look at what some of these early euprimate brains look like, in some ways they already look kind of like a modern primate brain. So modern primates are very much visually directed animals. If you think about kind of how you perceive the world compared with, say, how your dog perceives the world, your dog is a nose-first animal, right? It will, the first kind of sensory modality that it tries to perceive the world with is smell. We do it with our eyes. That's the first thing that we approach any new situation with. And as a result, this part of the brain, the olfactory bulbs, which are related to smell, tend to be fairly small in primates. And this is true even of this 48 million-year-old euprimate. It's 1.4% is very low compared to other mammalian orders. But what we do have is we have an expanded cerebrum. And this is reflected in how round this brain is, because it has a well-developed temporal lobe, and also how far back the cerebrum goes here. And in particular, the part of the brain that we're really most interested in when we're thinking about, smartness, whatever the heck that means, is a part of the brain called the neocortex. Now, the neocortex is the outer part of the cerebrum. If you look at a human brain, almost everything you're looking at is actually neocortex on the outside. And in fact, in a human brain, it makes up 90% of the cerebrum and 76% of the volume of the brain in total. It's enormously important in a human brain. Basically, it is that part of the brain that does all the good stuff. Things like speech, reasoning, processing of sensory information, all the things we sort of think of as higher brain functions. It doesn't do things like, breathing and maintaining basic body functions. It does all the sort of the things we're proud of about ourselves as humans. Now, because it's actually the outer surface of the brain, at least in relatively primitive forms where there isn't a lot of this sort of infolding that we get in a human brain, you can actually get a fairly good estimate of the size of this part of the brain by looking at the surface area. So in 2015, my master's student Adam Long and a couple of us published a paper defining a way of quantifying the size of the neocortex, based again on work done by Harry Jerison. He did it, but he never explained how, so we actually explained how to do it. And basically it involves using anatomical landmarks to section out that part of the brain that would have been the neocortex, and then comparing that with the surface area of the rest of the brain. And this is actually Jerison's data, and what he showed is, again, euprimates have always been exceptional for their time period. So these are the euprimate points here. This is sort of an undifferentiated mass of other mammals. We're actually going to come back to that point. But compared to this sort of undifferentiated mass of other mammals, euprimates have larger neocortices than other groups. We're very smart. Okay. So that's what we know from euprimates. But the problem is, to some degree, they're already too specialized. So the analogy that I kind of like here is, if you want to study human evolution, and you want to study bipedality, walking around on two legs, you can't only study modern humans, because we already do it. We're already pretty good at it, right? You have to study the things that weren't as good at it yet, that were in the process of evolving that characteristic. So in the same way, if we want to study primate origins, we can't only study the things that look like modern primates. We also have to study the things that in some ways don't look exactly like modern primates, that are in the process of evolving these features. And again, our argument is that these guys, these Plesidapiforms or stem primates, constitute that group. We have Endocasts for 4 families. Well, I know we have 4 families, and I'm going to tell you guys today about 4 families. Actually, only 3 of them are published, so you're going to get to see some unpublished data today. So this is the first one we published. I already showed it to you, Ignatius grabullianus. We published it in 2009. And you can see now, since you know how to read EQs, there's actually 2 ways of calculating them, but no matter how you calculate it, they're well under 1, so wasn't the smartest kid. We actually have a second specimen, which my lab produced the Endocast for a couple of years ago now. We still haven't published it. Interestingly, they're actually quite different in size. So in terms of absolute volume, you can see this is cubic centimeters. So this is about the size of a walnut, okay? They're teeny tiny. This is even teeny tinier, so this is less than 2 cubic centimeters. But because it was actually a smaller animal, its EQ is quite similar. Now, you might wonder why I'm going on about that. As paleontologists, when we have a sample size of more than 1, we're always excited. So we have 2, yay! Okay. The second one we published in 2010 was this one. This is a critter called Microcyops annectens. It's actually a little bit bigger. You can see this is 5 millimeters here, so it's not quite as tiny an animal. We actually had a natural endocast of the same species from the same site, which again, sample size of 2, very exciting. And this is a partial natural endocast of a slightly earlier occurring species of the same genus. Now, just literally like last week, my master's student, Chelsea Mikowski, completed an endocast for another member of this family. It's quite a smaller animal. So this scale actually applies to this endocast. You can see that's 5 millimeters. Okay, so this skull is very similar in both size and shape to a gumball. It's very small. It was a specimen that came out of a limestone, but it was what my postdoctoral supervisor, Alan Walker, used to call Irish stew. So it had all kinds of pieces of bone in different places, all kinds of messed up. What she had to do is actually take each of those individual pieces of bone and separate them out and then sort of jigsaw it back together, which took about a year and a half. But the end result actually is a fairly complete endocast. And I didn't put any numbers down here because we haven't actually finalized the numbers for this specimen yet. But what's interesting about it is its EQ is not that different from other Plesidaebriforms, but it actually has quite large olfactory bulbs, which is interesting. Okay, the third family, and this is the one that you guys are getting a sneak preview of something that's not published yet. This is the skull and reconstruction of a critter called Carpalstimpsoni. It's interesting because it's the only Plesidaebriform for which we know it had a grasping big toe, which is actually a primate-like feature. So it had like a nail and was able to actually grasp around a branch. That's different from things like, say, a squirrel that actually can't do that. They don't have an opposable thumb or big toe. Again, this specimen very much like Irish stew, but my master's student Rachel Rusin actually took again all these pieces separately, reconstructed them, and it's a little bit messy, but actually if we mirror image the good half, we get reasonably good estimates. It was slightly disappointing because we thought since this animal was more behaviorally like a euprimate, it might have a larger brain. Not so much, actually. Oh well. And then the fourth family is Plesidaepidae. This is work done by other research groups. So this is the fourth family of Plesidaebriforms. So of the 11, we now have endocasts for 4 of them, which is definitely a lot better than 10 years ago. So when we plop that data into our bar graph here, what we can see is that they tend to generally be smaller in terms of their brain size than fossil euprimates. So these stem primates don't show that sort of increase that we're inferring fossil euprimates to have experienced. And if we look at the form of the brain, they actually have pretty big olfactory bulbs. So that suggests these were animals that were more nose forward than a living primate. They were more like your dog, perhaps, than like us. And very limited development of the cerebrum. So this is actually a part of the brain called the midbrain. Normally, you can't see it on endocasts because it's covered by the cerebrum. In this case, it's not. So this has not expanded that part of the brain. In terms of trying to figure out what that means, we have to go to modern models because we can't look at the function of parts of the brain in a fossil because the fossil's dead. No matter what you do, it's not going to get up around and start dancing, right? So this is a functional map of the brain in a macaque monkey. And what you can see is there's a lot of the brain that's actually devoted to visual processing. Actually, 50% of the neocortex in macaques is given over to visual processing. And in particular, the back part of the brain and the temporal lobe is very heavily dedicated to visual processing. Well, actually, those are exactly the same areas that are underdeveloped in this Plesidaebriform. We have a very short cerebrum, so it's not covering these caudal colliculi, and we don't get that sort of round shape that we get because of the development of the temporal lobe. So that would suggest that what's different about these stem primates is that they weren't as visually oriented. Now, what's really interesting about that, I think, is that these were animals that were already behaving in many ways like primates. They were arboreal, they ate fruit and bugs, but they were doing those things with actually quite a different set of senses than a modern primate. They were much more oriented towards smell and much less oriented toward vision. And that's very interesting to me because it means that they were living actually quite different lives than the modern primates that we might want to use as their models. We also calculated neocortical area. So most of this data is actually Jerrison's data. So these are the U primate points here. And what you can see is the Plesidae are all sort of more typical mammals. But there's a problem with this graph. And I say that even though I was a co-author on the paper that published it. The problem with this graph is what we're basically doing here is we're comparing these primates against a whole bunch of other mammals, just other mammals, right? The problem is that the other mammals in this sample are largely things that aren't particularly closely related to primates. They're mostly ungulates, so things related to, say, cows and horses, and carnivores, so animals related to cats and dogs. We're not particularly closely related to ungulates or carnivores. So a lot of these blue circles represent animals that are really of only peripheral relevance in terms of making comparisons to primates. And what that means is, okay, yes, compared to this sample, they look kind of typical, but this sample might not be telling us about what actually was primitive at a deeper level for primates. So interpreting this then becomes a little bit questionable. So the answer to that is to start looking at some animals that are more closely related to primates. And as I mentioned, really difficult to do that with these guys because they're so poorly known from the fossil record. But gliries are actually quite well known. And in particular, there are a lot of fossil rodents. One of the really nice things about having graduate students is that they get you excited about things. I really wasn't that interested in rodents, I have to admit. To me, they're kind of vermin. But it turns out rodents are fabulous. There are over 2,000 species of rodents. Okay, so in comparison, depending on what phylogeny you use, there's maybe 500 to 700 species of living primates. There's a lot more rodents. And they're much more diverse ecologically, actually. So you have two different rodent groups that evolved gliding. You have rodents that are, semi-aquatic. You've got ones that hop around on the ground. You have ones that live in the trees. You have a fair diversity in terms of size. You go from, mouse-sized critters up to the capybara. The capybara weighs about 70 kilos. Which is a little bit bigger than, say, a female chimpanzee. So not quite as much size diversity among living rodents as among, say, living primates. We don't have any living rodents that are as big as, say, a male gorilla. But actually, there are fossil rodents that were even bigger. There was a fossil rodent known from South America that weighed about 700 kilograms. That is 4 times the size of a living modern gorilla. So that is a big rodent. We haven't studied that guy, unfortunately. I would love to. But so rodents are fantastic, and they open up all kinds of possibilities of studying ecological variation and all this kind of stuff. And it turns out that rodents actually have a pretty decent fossil record, particularly from our perspective in terms of crania in collections. So what we've been doing is going into collections and micro-CT scanning rodent skulls. And we've built actually a fairly good sample, but I'm going to just talk about a few of the early ones that we're interested in today. So this specimen here is a species called Ascaramys typus. It's from the Oligocene, so it's not really, really old. It's not as old as some of these old fossil primates. But it's actually quite a common species. And as I was going through the galleries yesterday, you actually have a really nice specimen of Ascaramys typus. It's actually a partial skeleton with a skull. So go and take a look, because it actually would have made a lovely endocast. And there's actually a lot of Ascaramys typus in collections because it comes from a time period when there's a good fossil record for mammals. And that was actually the first one we did, well, by which I mean my first one my PhD student Ornella Beltran did with my help. And we actually presented this at various conferences as the first virtual endocast for a fossil rodent. But because of the vicissitudes of publishing, actually it's not the first published virtual endocast for a fossil rodent. That goes to these two specimens here. So this is a critter called Paramys delicatus. It's rather ironically named because by rodent standards it was actually quite large. It was about the size of a sick cat, so sort of 6 pounds-ish. So if you have a cat that hasn't been doing too well, that's about the size that this rodent was. And then Paramys copi was about a kilo, so a bit smaller. And actually just last week, in Proceedings of the Royal Society Part B, we published the endocasts of these guys. So these are actually the first published virtual endocasts of fossil rodents. In terms of size, we tend to think that we're smarter than rodents, I guess. I made an allusion to that earlier, assuming that we're smarter than mice. Well, when we calculate their EQ, actually, these early rodents are not that dumb. They actually have much higher EQs than we had anticipated. They're actually within the range of Plesiodapiforms and even within the range of some primitive fossil euprimates. So their brains were not particularly small. And if we actually look at the form of the brain, they look terrifyingly like stem primates. They have a lot of the same features, which maybe we're just getting at something that's primitive for this entire group. So for example, they also have fairly large olfactory bulbs. They also have, although you can't see the caudal colliculi, they do have a patch of midbrain here, which means that the cerebrum was relatively short. Fundamentally, we're maybe starting to get at some kind of primitive model for this entire group that includes both primates and rodents. However, the one thing that is really distinctively different is the size of the neocortex. And again, this is the part of the brain that if we want to think about sort of smartness as being some meaningful thing, is really the part of the brain we should be most interested in. So in most Plesiodapiforms, we have a percentage of the overall surface area that counts for about a quarter of the neocortex, about a quarter to a fifth, depending on which individual you look at. So they're all well over 20%. Whereas both of the fossil rodents we did are somewhere around 16 to 17%. That's actually really low. And that suggests that there must be other parts of the brain that are larger in these rodents compared to stem primates, because again, in terms of overall size, they're not that small. Now, we're not really sure what parts of the brains are. We know they're not the olfactory bulbs because we can quantify that. It's probably parts of the brain that we're not particularly good at quantifying, like this part, the cerebellum here, which you can see seems to be sticking up higher. We're not really sure how to put a number on that, so that's kind of one of the things that we're studying. So if we come back to this plot, okay, compared to this undifferentiated mass of random ungulates and carnivores, stem primates don't look that impressive. But the fossil rodents are actually down around here. So to the extent that that's relevant for telling us what's primitive for primates, maybe all primates, including these primitive stem primates, had actually expanded their neocortex. We're already starting to get smarter, even before we come to the common ancestor of all living primates. Okay, so just to come back to our three questions here, in terms of the first question, one of the really key points in starting to increase primate brain size seems to be related to the evolution of vision. This sort of transition from something that looks like a stem primate to something that looks like a primitive euprimate seems to be tied to parts of the brain that specifically related to vision. And that's actually supported by other contrasts between these groups. So something that all euprimates have is we actually have a bar of bone behind our eye called a postorbital bar. It's thought to help stabilize your eye so that it improves your vision. Well, all euprimates have this, stem primates don't. So there are other sort of elements of the skull which suggest that what was really key in terms of that sort of transition from a stem primate to a euprimate might be related to improvements to visual processing and becoming a more visual animal. In terms of when that happened, the earliest euprimates appear about 55 million years ago. So as early as 55 million years ago, we're starting to see the first stages in primate brain size increase. Now remember that they still don't have big brains compared to living primates, so there's many additional kind of parts to the story that we need to understand. That didn't get us to a completely modern primate brain. But what that gets us is starting to move in the direction that's going to ultimately lead to this distinctive characteristic of primates in having large brains. It turns out that the most difficult question to answer seems to be this last one. And we've been looking at fossil rodents as one model for what's primitive for the primate brain, but they're not the only possible model. And so we need to start thinking about a variety of other possible pictures of what might be primitive at a deeper level for the primate order. But our first indication from this primate record suggests that actually maybe what's really interesting is not so much brain size per se, but evolution of the parts of the brain that are the ones that we're most interested in. So maybe what we really need to be thinking about is not just brain size evolution, but specifically neocortical evolution. And if our story that we just told in a paper that just got published is true, it's possible that increases in the neocortex actually started earlier than increases in other parts of the brain. So it could be that the stem primates that we thought were kind of primitive looking in their brain actually had, compared to their ancestors, expansions in the neocortex. And that's an interesting possibility. I'm not sure that I believe it yet. But what's really peculiar about that is we have no idea what would be driving that. These are not animals that would be, for example, particularly visually oriented, so it's not the same explanation. We need some other explanation that we honestly haven't come up with yet for why these very earliest primates would need to be smarter than these very early rodents. Okay, I'd like to just finish up by thanking everybody, and in particular, my collaborators, many of whom are students. A lot of this work is being done working with graduate students, working with undergrads who come into my lab as technicians, and they really do a lot of the grunt work that makes this story be a story rather than one picture that I could show you guys. And so thank you all as well for coming today. I really appreciate it, and I think I can take questions. Can I take questions? Yeah, I'd be happy to take questions.